Hey everyone, my name is John Nahan. I am the manager of Energy and Earth Sciences here at the St. Louis Science Center, and I'm here today to show you around our new stage space. Hopefully you've gotten a chance to check it out. It's been open both before we shut down and after we've reopened, so come on by. We have about 4 to 5 shows every single day. You can check out our schedule online if you would like to do so. Once again, please come visit us, but let's go ahead and take a look at what it's like to be up on stage and back in our green room. My personal favorite part about this new stage is all of the safety features that we have added. So there's 2 main things that I want you to notice as we come up here. One are these really awesome plexiglass barriers. Now, these barriers are super essential for keeping us safe and keeping you safe as well, because we have a lot of different hazardous chemicals and materials that we use up here. And we want to make sure that we can keep that little bit of separation, but it still allows us to get really close and personal and make that science feel accessible. Additionally, we have this brand new, really fantastic eye washing station. We've never had to use it so far, but it's really important to have something like that around in case something does go awry, and we can respond to emergency situations in the way that we need to. Now, my second favorite thing about the new stage space is all of the audio-visual capabilities that we've got. So we have different cameras sitting around from a bunch of different angles, which you'll get to see in a little bit here as we record Germbusters. We also have the capacity to not only record, but do virtual streaming of people like different science experts to bring them to our audiences here, or somebody who maybe can't make it, but we really had them fit into a specific topic we're talking about. So we're really excited about all of that audio-visual capability that we've got going on, as well as just like the fun lighting and stuff that we get to do. A lot of that can be controlled from up here. However, we also have the ability to control everything from our booth, which is sitting back here, way back in that corner there. You'll notice we can put somebody back there. They have the ability to change camera angles, to change the lighting that we have going on, to play different sounds or PowerPoints or whatever you need. It's really opened up our capabilities to provide a lot more content here. Something else that I love about this stage is the ability to see everybody in the audience. So you'll notice that I, even from here with all this stuff in the way, have an angle to look at every single person who's sitting in the audience. And that's really helpful for me as an educator and as somebody who's up here performing, as well as the other people who do the same thing, because we can gauge your reaction as to how you're feeling about the show or what's really connecting and clicking with people. And that's one of our main goals, is to make science accessible for everybody. So if we notice that somebody is really excited about something, or if we're kind of losing people, we can adjust our show to make sure that you're receiving the content in a way that makes you feel connected to it, and make sure that you're learning still. All right, so let's go ahead and check out the green room back here. This is where we prepare all of our shows. And it's where we house a lot of our materials as well. So I'm gonna go ahead and open this up for us, and we're gonna come on through. Now, once again, my favorite part about this is our expanded space to be able to have more PPE, which is personal protective equipment, as well as some fire extinguishers sitting around and different things that really up our level of safety. Now, it may look like a random hodgepodge of stuff that's in here, and that's kind of because it is. And that's one of the ways that we, once again, try and make science accessible for you all, is by using materials and things that feel relatable to you and things that you recognize. So we want science to feel like you can be connected with it, and try and have a little bit more fun than maybe you get to in a traditional classroom. So once again, we've got all this really awesome stuff here. That's a big old Tesla coil back here. We've got our Van der Graaf generator, Jacob's ladder, Rubens tube. Yeah, there's all sorts of stuff in here, and it's packed away really tight and really well. But now that I mentioned the AV booth a little bit, let's go ahead and check it out and see what capabilities it has to bring science better to you. So in the booth, we have a whole bunch of different stuff that allows us to really be super versatile with what we do on the stage. We have a switcher that controls video, we have a switcher that controls audio, and we have something that controls lighting as well. As I mentioned previously, this allows us to do really awesome recordings and also have virtual speakers come in and talk to us on the stage. But the person in the booth has the ability to control so much more. So if we're having a live speaker, they can actively change the lighting, they can actively change audio levels. All of this allows us to have all sorts of different presentation types be covered, whether it's a speaker, whether it is some type of performance like a skit or a comedy. It also allows us to have live bands on stage as well as live streaming. Essentially, it allows us to do whatever we want to. All right, now that we've done a quick behind the scenes tour, we're gonna go ahead and get our show Germbusters started. So first things first, most important, gonna go ahead and grab my lab coat. get that put on, and I'll meet you on the stage. All right, everybody, now that we're all geared up and ready to go, we're gonna talk about viruses. But before we do that, why don't we light some stuff on fire? So over here, I've got this little denatured alcohol torch and some different spray bottles that are filled with denatured alcohol and some other chemicals. Now, as we turn the lights down a little bit, what we're going to do is spray this denatured alcohol over this torch, and we're gonna get some fire. This is our lithium chloride mixed with denatured alcohol. That's why it kind of has that nice pink color. Now, what I want you to notice is the shape of the flame as it goes forward, all right? Think if it starts small and gets bigger, if it starts big and gets smaller, if it makes a square, if it makes a circle. Just think about the shape as it's coming out. This is our strontium chloride mixed with our denatured alcohol. We're gonna do it again, and once again, I want you to look at that shape. And then last but definitely not least is our copper chloride here. So by now, what you've probably noticed is that it gets bigger as it moves forward, right? It makes the shape of kind of a cone here. Now what's important about that is that's how a sneeze travels as well. That's how sneezes, coughs, talking loudly, anytime you're ejecting particles from your mouth, they start small and they get big. That's because of the fact that there's air out there that's pushing back against it and making it spread out. Sneezes, interestingly, can go anywhere from 23 to 28 feet. So let's do this one more time, and then we're gonna start talking a little bit about what viruses are. All right, I will put this back. We're gonna get that put there. Awesome. Now, we're gonna come over here to my blackboard, and we're gonna take a look at this. This is a depiction of a virus. Now, viruses are a really interesting biological entity that act as infectious agents. Viruses are really, really teeny. They are submicroscopic. They're way smaller than bacteria. They're very different from bacteria as well. And not only are they small, but they're also really, really simple. And we're going to talk about that in just a second. Now, viruses infect literally every biological entity on the planet. They infect plants, they infect animals, they infect bacteria, they even infect archaea. So viruses are able to get into everything, but we don't even really know if we can consider them alive. Some biologists say that they are alive, others don't, but that's because they exhibit some signs of life, but they don't exhibit others. The two main ones that they're missing is the lack of ability to metabolize stuff, which is take food and create energy out of it, and then also to replicate on their own. They have to have a host in order to make more of themselves. So now that we've gotten that out of the way, we're going to talk a little bit about what viruses are made of. There are two main components and sometimes a third. The two main components to start are genetic material and protein capsids. So the genetic material is right here, represented in red and blue. In the case of SARS-CoV-2, the genetic material is single-stranded RNA. This is the stuff that goes into your cells or the cells of whatever host the virus is trying to infect, and it tells your cells, hey, make some more viruses. So it's able to hijack some of your organelles and other proteins in order to create more viruses, to create more proteins as well. This protein capsid, this green line right here, is essentially just a case that holds in that genetic material. And that's it. That's all that viruses are usually made out of. Sometimes we have a third thing, which is a lipid envelope. In the case of SARS-CoV-2, there is a lipid envelope. A lipid is a type of fat, and that's on the outside right here. So that's all that a virus is made of. They're really, really small. We've got these three components. Once again, our genetic material is what goes in and tells those host cells to make more viruses. The protein capsid helps hold it in, and then sometimes there are lipid envelopes as well. In addition, there are some other spike proteins and other things that are associated with viruses that help them get the job done. But for the most part, it's just these two or three things that we've got that make up a virus. And they're everywhere. They're absolutely everywhere. There are more viruses by number than every other type of biological entity combined. You right now probably have many viruses on you, but most of them are really harmless and not able to infect humans. They're very specific to their host cells. All right, so we're gonna go ahead and move back on over here and talk a little bit about masks. So I want you to take a look at this little diagram that we've got going on here, and what you're gonna see is kind of three main things. We've got a face over here, we have some particles over here, and then we have this white stuff here, which is going to represent our mask fibers. Now, I don't want you to think of your mask when you're wearing it as a colander or a strainer or anything like that. I want you to think of it as a spider web. So the whole point of the mask is to get these particles, which could potentially be carrying coronavirus or other illnesses, to touch one of these fibers. And once it does so, it grabs them and it holds on to them really tight. Now, the reason I have 3 here is because we classify droplets that exit from our mouth and other droplets in the air as well, kind of in these 3 main categories of large, medium, and small. And we categorize them that way because they move differently as they move through the air based on their size. So our largest ones, around 1 micrometer, get stopped really easily by these fibers. As you breathe in, there's a bunch of air rushing towards your face, coming through this mask. This large particle will meet its end pretty quickly as it touches one of these mask fibers. This smaller one down here, even though it seems like it might get through the mask really, really easily, it actually has a really hard time, and that's because of something called Brownian motion. As it moves towards your face, there's a whole bunch of air molecules that, because it's so small, it can knock around a whole bunch. So it's going to try and go through your mask, and it's going to hit here and here and here and here and do a bunch of random movements. And a lot of the time, as it goes through your mask, it's also exhibiting Brownian motion and very easily sticks to one of those fibers. The ones that are really difficult for us to deal with are these medium-sized ones, around 0.3 microns. That's because they can move with the air and generally flow through these mask fibers as the air comes towards your face. Now, we deal with that in 2 ways. One is by putting a bunch of layers on your face or over your face. Multi-layered masks do a really good job with these medium-sized particles, and things that have compressed fibers rather than woven ones also work really well. So the whole idea is that as they're moving through the mask, we're either getting them to stick through electrostatic interaction, kind of almost like a little electric magnet to grab to the fibers, or by something called inertial impaction, which is almost like if you're driving a car and going around a tight corner way too fast, you're going to fly off the road. That's what we're trying to do these particles. They come in, they move a little bit too quickly as they're coming around one of these curves, and they accidentally hit one of these fibers and get stuck. So that's the way that masks work. They're highly effective, especially if they have those double layers, and they do a really good job of protecting other people as well and stopping all of those droplets from getting out into the air. So it's highly important that you wear your mask. One way you can tell if it's generally pretty effective or not is by taking it and holding it up to a light source. And if you can see light through it pretty easily, then you know you probably need a little bit of a thicker mask. So we're going to move on, and we're going to talk really quickly about how we kill viruses next. So there are 3 main ways that we kill viruses. One is with soap and water, washing our hands really well. Another is with ethanol or some other type of safe-to-use alcohol. And then the third is generally with a peroxide. So whenever we use soap and water, the whole point of what we're doing is to try and break down this outer membrane right here. So let's go ahead and do that really quickly. So this lipid membrane is crucial to the virus's functionality. So if we can break that down and get it to chunk into a bunch of little pieces, then the virus really becomes super ineffective. Also, the mechanical action of our hands helps us do this. So I'm going to go ahead and do some mechanical action with our hands. There we go. And our virus is gone. So soap and water is incredibly effective. It does work really, truly well. Now here at the Science Center, we use a peroxide-based cleaner. Peroxides work by releasing a whole bunch of oxygen, which is really, really greedy. It likes to take other substances and rip them apart. So this oxygen-based cleaner has peroxides in it and then a little bit of a surfactant as well, kind of like a soap to help pick up dirt and stuff and get it out. So I'm going to show you what happens whenever we release a whole bunch of oxygen at once. This experiment is called elephant toothpaste. Inside of here I've got some food coloring along with some soap. I'm going to pour in sodium iodide, which is a type of catalyst. It's going to help that oxygen push off of that molecule really, really easily. And then I'm going to put in some hydrogen peroxide, which is where our peroxide's coming from. The chemical formula of this is H2O2, so basically water with an extra oxygen on it. And like I said, that oxygen really, really wants to come off quickly. So we're going to go ahead and pour it in there and see what happens. And we got elephant toothpaste. That was a good one. So what you'll notice is that we've got a whole bunch of steam coming off of the top. Now that steam is quite a lot of oxygen and heat that's being created from this exothermic reaction. Like I said, these peroxides are really good at ripping things apart because there's so much extra oxygen around whenever we pour them on something. So we're going to go ahead and move over here and talk about the last type of sanitizer that we use, which is alcohol. All right, so now that we have talked about how we use peroxides and how we use soap and water, I want to talk to you a little bit about how we use alcohol in order to kill viruses. But while we do so, I figure we might as well blow something up. So I'm going to take some denatured alcohol, which is a mixture of ethanol and methanol. I'm going to pour it into our whoosh bottle. Although, you know, I think today we might as well use a whole lot. So let me just go ahead and get this screwed off here. All right, put that down. And let's just pour it in here. There we go. I think that should be more than enough. I'm going to set this over here really quickly. Awesome. Okay, so the way that we use ethanol and other alcohols in order to kill viruses is essentially by destabilizing them. Whenever we put alcohol surrounding a virus, we're taking away the stability that dry air and moisture can add to a virus. So that whenever there's water around it, it really has a good ability to stick together and stay super stable. But whenever we put that alcohol around it, it loses that. And on top of that, we rub our hands together, which then breaks that virus into a bunch of little pieces. Now, something that's super important to know about ethanol-based hand sanitizers is that you really want to aim for somewhere between 60% and 80%. Under 60, and it won't be high enough of a concentration to work. Over 80%, and it'll actually evaporate too quickly in order to be effective as well. So we have ethanol inside of here. It's also super flammable, but it's mixed with methanol, so it's not a really good disinfectant because it can actually be pretty toxic to humans if used unsafely. So we're gonna go ahead and light this on fire. I'm gonna get it put in here, moved over to this side, and then we'll get it going. Now this is pretty loud, so I'm gonna go ahead and put these on and count us down. Here we go, in 3, 2, 1. Thank you for listening to everything that we had to say today and getting to see Germbusters while you're at home. Once again, I'm John Nahan, the manager of Energy and Earth Sciences. If you have any questions about anything we've talked about today, whether it is Germbusters or whether it is about the new energy stage itself, please feel free to reach out and we'll get back to you as soon as we possibly can. And once again, thanks for joining us for this member mission. We'll see you soon.